So imagine a world where treating bacterial infections is as simple as taking a snapshot. So this might sound like something that's within the realms of science fiction, yet it's actually closer to reality than you may think. And this is because of the innovative work of a group of dedicated researchers. So welcome back to the Chem Vibes, your go-to channel for all things chemistry. And today I want to be starting a new series known as Simplifying Chemistry Research. And we're going to be starting off with research that involves creating probes that can target the detection of bacterial infections, specifically Staphylococcus aureus. Staphylococcus aureus is a harmful bacteria or pathogen that causes many types of infections in humans. So these infections can be as minor as skin infections or as serious as heart infections and dangerous bloodstream infections. So Staphylococcus aureus often forms a protective layer that's called a biofilm in the body, and this makes it harder to detect and treat. So therefore, there is a need for tools that can specifically visualize or image these biofilms. Okay, so the research team journey started off by first identifying enzymes. And enzymes are like little machineries that exist in all living systems and living things. And what they do is that they carry out functions that helps the living thing to grow and to exist in the world, if that makes sense. So they found this enzyme that exists in the Staphylococcus aureus, and it's known as FPHE, and this enzyme, it is specific or unique to this pathogen because it wasn't found in other bacterial counterparts. And from there they decided that maybe they could find a chemical structure that could specifically bind to this enzyme FPHE. So the researchers they discovered a chemical structure or a chemical compound that's called oxadiazolone, and they found that this structure could act as a probe and bind specifically to the FPHE enzyme that's found within the pathogen. So to confirm all of this, they use a technique known as X-ray crystallography, and this is just a technique that allows them to see like molecular structures as to how the enzyme is binding with the chemical structure. So the probe itself is an ingenious design and it's consisting of three main parts. So first there is the warhead which connects directly to the enzyme. Then there is the linker portion, and the linker portion connects the warhead to a fluorophore. So a fluorophore is a dye molecule that glows brightly when exposed to light, and this makes it possible to track the molecule for imaging studies. The research team then wanted to ensure that the probe wouldn't harm the bacteria, because after all, the aim was just to visualize the bacteria and not necessarily kill it. So some experiments that they did show that in high amounts, the probe didn't hinder the bacterial growth. And therefore, this indicated that it binds specifically to the FPHE enzyme without disrupting any crucial bacterial functions. So, with the safety of the probe established, the researchers then move on to see if they were able to visualize FPHE in living bacterial cells. So, using a special microscope, they were able to see a glow in bacterial cells that signified the presence of the FPHE enzyme. So they then wanted to test if the probe could stick only to S. aureus bacteria when in the presence of other type of bacteria, which would also include harmful gram-positive and gram-negative bacteria, as well as yeast. So when they looked at the labeled cells under the microscope, they found that the probe mostly stuck to FPHE in the Staphylococcus cells. So in further tests, they injected the probe into mice that were infected with Staphylococcus and imaged them using the special microscope. However, to compare, they injected mice with saline and they injected others with E. coli. And what they observed was that the probe gathered only at the sites of Staphylococcus infection, and it didn't gather at sites of saline or E. coli injection. And the amount of fluorescence or glow that they saw at the infection sites was significantly higher than the non-infection sites. So this indicated the probe's ability to specifically target Staphylococcus aureus infections. This type of research represents a breakthrough and a beacon of hope for all those who are affected by these type of infections. So share your thoughts in the comments below. Don't forget to like, click and share. Thanks for watching. Until next time, stay curious.